Welcome back, everyone, to another deep dive. This time we're going deep into the world of biochemistry.
Oh, fun.
Yeah, so specifically we're focusing on gluconeogenesis.
Okay.
Imagine this scenario. You haven't had any carbs in a while,
Right.
but your brain still needs glucose to function. Where does that glucose come from?
Yeah, that's where gluconeogenesis comes in.
Exactly. It's like our body's backup generator.
I like that.
Yeah, turning non-carbohydrate sources into glucose.
It is a fascinating process, for sure.
It is. And the source material we're using today is an excerpt from a biochemistry textbook.
Great.
Yeah, so if you have a solid foundation in biology, think maybe second year biochemistry level.
Perfect.
This will be right up your alley.
Yeah, this excerpt dives deep into the substrates
Okay.
for gluconeogenesis.
They're like the raw ingredients.
Yes, exactly. The raw materials that get transformed into glucose.
Cool. So we're breaking down the ingredients list for making glucose
Precisely.
when carbs aren't around.
Okay.
Got it.
Okay.
So let's start with a familiar substrate, lactate.
Sure.
You know, that burning feeling you get in your muscles during intense exercise.
Oh, yeah, definitely.
Part of that's due to lactate buildup.
Right.
So what happens to all that lactate?
Well, it actually plays a crucial role in the Cori cycle,
Okay.
which is this really cool interplay between your muscles and your liver.
Okay.
So during strenuous exercise, your muscles produce lactate,
Uh-huh.
then it travels to the liver,
Okay.
and in the liver, it gets converted back to glucose through gluconeogenesis.
Oh, wow. So it's like a round trip.
Exactly.
Lactate goes to the liver, turns into glucose.
Yep,
And then glucose comes back to the muscles.
exactly. And that recycled glucose can then be used to replenish glycogen stores in the muscle or released into the bloodstream to maintain blood glucose levels.
Wow. That's pretty amazing.
Hmm.
Like our bodies are so efficient.
They really are. It's a beautiful system.
Yeah. Okay, so we talked about lactate. What about other substrates that can be used to make glucose?
Well, amino acids are a big one.
Oh, okay. The building blocks of proteins.
Exactly. And a lot of them can be converted into glucose.
So if we really need it, our body can break down protein to get those glucogenic amino acids.
That's right.
Yeah.
Especially during periods of fasting or starvation.
Interesting. So it's like sacrificing muscle mass to fuel the brain and other vital organs.
Precisely. It's a survival mechanism.
Wow. But not all amino acids can do this right.
Right. There are a couple of exceptions.
Okay, like
Leucine and lysine.
leucine and lysine. Got it.
They can't be converted into glucose.
Okay, so those are out. What about fats?
Fats.
Yeah, they're a major energy source.
They are. But they're not great gluconeogenic substrates.
Really? So even though we store so much energy as fat, we can't easily turn it into glucose.
It's not that straightforward.
Okay, why is that?
Well, it comes down to the metabolic pathways involved.
Huh?
When you break down fatty acids, you get acetyl-CoA.
Okay.
And acetyl-CoA can't be converted to glucose in animals.
So it's like a dead end in the pathway.
Pretty much. But there is one exception.
Oh, it's
Glycerol.
glycerol. Okay.
Yeah, it's a component of triglycerides.
Right, the fats we store.
Exactly.
Yeah.
And glycerol can enter gluconeogenesis.
Oh, interesting. So glycerol has a special pass.
You could say that it can be converted into dihydroxyacetone phosphate, which is a key intermediate in both glycolysis and gluconeogenesis.
Okay, so it can jump onto the gluconeogenesis pathway.
Exactly.
That's cool. So most fats can't directly become glucose, but glycerol can.
Right.
Okay, I think earlier we briefly mentioned propionate,
We did.
and it's particularly important for like cows and other ruminants.
Yes, propionate is crucial for them. It's a 3-carbon molecule produced in their rumen during fermentation.
So their digestive system is churning out this gluconeogenic substrate.
It is, but the conversion to glucose is a bit indirect.
Oh, wait, how so?
Well, first propionate is converted to propenyl-CoA
Uh-huh.
and then to secanyl-CoA,
Okay.
which is an intermediate in the citric acid cycle.
Oh, right. So it kind of hitches a ride on that cycle?
Exactly.
Yeah.
But even though the citric acid cycle is usually about breaking things down for energy,
Yeah.
some of its intermediates, like succinyl-CoA, can be used for other things, like gluconeogenesis.
So it's like a metabolic roundabout.
I like that analogy.
Molecules can hop on or off depending on what the body needs.
Exactly.
That's really cool. But getting from propionate all the way to glucose must involve a bunch of steps.
It does. From succinyl-CoA, you need a few more steps to get to oxaloacetate.
And that's a key precursor for gluconeogenesis.
Precisely.
Wow, this is a really complex system.
It is. It highlights the elegance and complexity of these pathways.
It does. Okay, so we've talked about molecules that can be used to make glucose.
Yes.
Are there things that can inhibit gluconeogenesis?
Yes. Definitely. Well, ethanol is a big one.
Ethanol like alcohol.
Yes. When ethanol is metabolized in the liver, it increases the NADH to NAD plus ratio.
Right.
And that messes up the balance of reactions needed for gluconeogenesis.
Also makes it less efficient.
Exactly. This can lead to decreased glucose production and potentially even hypoglycemia.
Oh, wow. So giving alcohol to someone with hypothermia is actually a bad idea.
It is. It can actually worsen the situation.
That's good to know. It busts that myth wide open.
Definitely. So even though our bodies can adapt and use different fuel sources,
Yeah,
there are limits.
that makes sense.
Yeah.
Okay, so we've covered the different substrates that can be used to make glucose through gluconeogenesis.
We have.
But how does our body actually regulate this whole process?
That's a great question.
How does it know when to ramp up gluconeogenesis and which substrates to use?
That's where things get even more interesting.
Okay, so maybe that's a good place to pause for now.
Yes, there are.
We've covered a lot of ground today, from lactate to glycerol to propionate.
And even busted a few myths along the way.
Exactly. It's clear that gluconeogenesis is a vital pathway.
Absolutely essential for survival.
But there's still so much more to explore.
There is. We've only just scratched the surface.
So stay tuned, because in our next deep dive, we'll delve deeper into the intricate enzymatic reactions and regulatory mechanisms that drive this amazing metabolic pathway.
It's going to be fascinating.
Definitely.
Welcome back to our deep dive into gluconeogenesis.
Back for more.
So last time we talked about the what of gluconeogenesis, like the different substrates.
The ingredients.
Exactly. Now we're going to tackle the how.
Oh, cool.
The enzymatic reactions that make it all happen.
Okay, so let's fire up those metabolic engines.
Let's do it. One of the most important steps is turning pyruvate into phosphenol pyruvate.
Phosphenol pyruvate, or PEP for short.
Exactly. It's a crucial step because it bypasses one of the irreversible reactions in glycolysis.
Oh, so we can't just run glycolysis backwards.
Not exactly. We need these special bypasses.
Makes sense. So how does this pyruvate to PEP bypass work?
It's a two-step process that starts in the mitochondria.
The powerhouses of the cell.
That's right. First, pyruvate is converted to oxaloacetate.
Okay.
And this reaction is done by an enzyme called pyruvate carboxylase.
Pyruvate carboxylase. Okay.
And it needs ATP, which shows you how much energy gluconeogenesis needs.
Like paying a toll to enter the gluconeogenesis highway.
Exactly. So now we've got oxaloacetate, but it can't just cross the mitochondrial membrane.
Right. It needs to get to the cytosol where the rest of gluconeogenesis happens.
Exactly. So oxaloacetate is converted to malate by mitochondrial malate dehydrogenase.
Another enzyme.
Yep. This reaction uses NADH, and it's reversible, so it's involved in both gluconeogenesis and the citric acid cycle.
Wow. So malate is our little traveler.
It is. It can exit the mitochondria and enter the cytosol.
Okay. And what happens once it's in the cytosol?
Malate gets converted back to oxaloacetate.
Back again.
Yeah. This time by cytosolic malate dehydrogenase.
So same molecule, different location.
Exactly. And this time it uses NAD plus and produces NADH.
Okay, so we're back to oxaloacetate, but now it's in the right place.
Right in the cytosol, where it can continue through gluconeogenesis.
So what's the next step in this molecular makeover?
Oxaloacetate is converted back to phosphenol pyruvate, or PEP.
Back to PP.
Yep. This reaction is done by the enzyme phosphenol pyruvate carboxykinase.
Say that 3 times fast.
I know, right? It's usually just called PPCK.
PPCK, much easier.
And this reaction needs GTP, another energy source, and it releases carbon dioxide.
So again, we're seeing that energy investment.
Absolutely.
Yeah.
Our bodies are willing to spend some cellular currency to get that glucose.
Makes sense.
Oh.
Okay, so we bypassed that first irreversible reaction.
We did, but there are 2 more to go.
Oh, bring on the metabolic detours.
The next one is converting fructose 1,6-bisphosphate to fructose 6-phosphate.
Okay, and remind me, in glycolysis, this step is done by...
Phosphofructokinase, or PFK.
Right, PFK.
Yeah.
But in gluconeogenesis.
A different enzyme takes over fructose 1,6-bisphosphatase.
Okay, so another example of a separate enzyme reversing a glycolysis step.
Exactly. And this enzyme just hydrolyzes the phosphate group off fructose 1,6-bisphosphate.
Okay, making fructose 6-phosphate.
And inorganic phosphate.
And this lets gluconeogenesis keep moving towards glucose. Got it. What about that last obstacle, going from glucose 6-phosphate to glucose?
Ah, that's where glucose 6-phosphatase comes in.
Glucose 6-phosphatase.
It's mainly found in the liver.
Makes sense, since the liver is all about managing blood glucose.
It is. But this enzyme is interesting because of where it's found.
Oh, okay. Where is it?
In the endoplasmic reticulum membrane,
The ER.
yeah, with its active site facing the lumen.
The inside of the ER.
Exactly. So glucose 6-phosphate has to be transported into the ER to meet up with glucose 6-phosphatase.
Like a secret meeting place for this reaction to happen.
And once it's there, glucose 6-phosphatase removes the phosphate group to make free glucose.
And that free glucose can then be released into the bloodstream.
Exactly, fueling the rest of the body.
So it's like a whole team of enzymes working together to make sure we have glucose.
It is. It's remarkable how they bypass those irreversible steps.
Yeah, and it's super efficient too.
It really is. But you might be wondering, why doesn't gluconeogenesis just run in reverse all the time?
Like, why not just reverse glycolysis?
That's a great question. And there's a good reason for it.
Okay, I'm listening.
It comes down to energy. Gluconeogenesis needs a lot of energy to make it work.
So it's not just about changing direction, it's about overcoming energy barriers.
Exactly. To make one glucose molecule from 2 pyruvate molecules, gluconeogenesis uses 4 ATPs, 2 GTPs, and 2 NADHs.
Wow, that's a lot of energy.
It is, which is why regulating gluconeogenesis is so important.
We don't want to waste energy.
Exactly. So the body has to carefully control when and how much gluconeogenesis is happening.
Like a delicate dance between glycolysis and gluconeogenesis.
It is a delicate balance. Now let's shift gears a bit and talk about the bigger picture.
Okay, sounds good.
The overall regulation of gluconeogenesis.
I'm ready. Let's see how it all fits together. Okay, so we've gone through these pathways and all these enzymatic steps of gluconeogenesis, but how does our body know when to ramp it up or slow it down?
Well, that's where hormones come in. They're like a control system,
Okay.
fine-tuning gluconeogenesis based on what our body needs.
So what are the key players in this system?
Insulin and glucagon. They're both produced by the pancreas.
So they're kind of like the metabolic traffic cops.
Exactly. They direct the flow of glucose.
I like that. So tell me more about how they work.
Okay, so when your blood sugar is high, like after you eat,
Uh-huh.
your body releases insulin.
Okay.
And insulin signals to your body, hey, we've got plenty of glucose, let's store it.
So insulin basically stops gluconeogenesis.
It does. It suppresses the enzymes involved in gluconeogenesis.
Like PPC.
Exactly. And it promotes glycogen synthesis.
Okay, so we're storing that glucose.
Right. But then when blood glucose levels drop,
Like when you're fasting or exercising.
exactly, then glucagon comes in.
So it does the opposite of insulin.
It does. It's like the counter-regulatory hormone.
So glucagon tells the liver, hey, we need more glucose, make it happen.
Exactly. It stimulates the production of those gluconeogenic enzymes.
And that promotes glycogen breakdown.
Right. So we release glucose back into the bloodstream.
It's amazing how these hormones work together to balance everything out.
It's a really elegant system. But hormones aren't the only factor.
Oh, right. What else plays a role?
Well, the substrates themselves, how much is available.
Okay, so like if you're on a low-carb diet.
Exactly. Your body has to rely more on gluconeogenesis.
Because it needs to make glucose from other sources.
Exactly, like amino acids from protein.
So our bodies are constantly adapting.
They are always adjusting to keep things balanced. But beyond hormones and substrate availability, there are other things that regulate gluconeogenesis.
Oh, like what?
Allosteric regulation and covalent modification of enzymes.
Those sound pretty complex.
They are, but I can try to explain them simply.
Okay, go for it.
Think of allosteric regulation like a volume knob on an enzyme.
Okay.
Certain molecules can bind to the enzyme and change its shape.
And that affects its activity.
Exactly.
Yeah.
It can either activate it or inhibit it.
So it's a way to fine-tune the enzyme's activity.
Right. Covalent modification is a bit different.
How so?
It's like attaching a sticky note to the enzyme,
Okay.
adding or removing chemical groups like phosphate groups.
And that changes how the enzyme works.
Exactly. It's another way to control enzyme function.
Wow. So we have all these layers of regulation working together to keep gluconeogenesis running smoothly.
It's really quite remarkable. But before we wrap up, there's one more thing I want to mention.
Okay, what's that?
The role of extrahepatic PPCK.
PPCK. I remember that enzyme.
It converts oxaloacetate to PP.
Right. One of those bypass steps.
Exactly. But here's the thing. PPCK isn't just in the liver.
Oh, really? Where else is it?
It's also in other tissues, like the kidneys and adipose tissue.
So other organs can do a bit of gluconeogenesis too.
That's right. Although not as much as the liver.
So what does that tell us?
It shows that gluconeogenesis isn't only about keeping your blood sugar stable.
Okay.
it's also involved in other things.
Like what?
Well, for example, it provides substrates for making triglycerides in adipose tissue.
So it's more than just a backup generator for glucose.
Exactly. It's a versatile pathway that's involved in lots of different processes.
Wow. So we've really come full circle here.
We have.
We started with the basics of gluconeogenesis.
Right. The different substrates.
And we've explored the intricate pathways,
The enzymes.
the regulation.
And even some of the broader roles of this amazing metabolic process.
It's been a fascinating deep dive into the world of biochemistry.
It has. And it really highlights how adaptable and complex our bodies are.
Exactly. So to our listeners, keep exploring and stay curious because the world of biochemistry is full of wonders waiting to be discovered.